We're moving into lab 6, and we're starting here. A good helpful textbook figure for distinct sections of a long bone, and we'll go right to that figure. So we have the terminology, the diaphysis, the epiphysis, the medullary cavity. So that starts here. The slender midsection of a long bone, that's the term diaphysis. And the enlarged ends, this entire side versus this opposite end. These are the epiphyses of a long bone, epiphysis singular. And with this section through the bone, cutting it open, we can see the hollow internal space known as a marrow cavity or medullary cavity. And finishing up with the superficial compact bone, and especially concentrated in the epiphyses, the spongy porous bone. And so we can see that here. Again, for a long bone, this femur, that's the concentration of much more of the spongy bone in the epiphyses, whereas much more of the compact bone, higher percentage seen here in the area of the diaphysis. But of course, it's all across at the surface. And just a closer detailed image of that. So we're looking here at the epiphysis with the, of course, spongy bone, and then moving out more closer to the superficial edge is where we see more of that compact bone. And here we're seeing the proximal end of the medullary cavity. So next we're moving on to the detail of osteon. We have a model here and then some detail. So we'll start here with this view of the osteon. Let's get a pointer. So this entire circular unit here, that is one osteon. And in the center of the osteon, with the blood vessels within, we're able to see the central canal. And let's continue from there on the list. So we have the lacunae, the lamellae, the osteocyte. So the lacunae are all these hollow cavities within the osteon. And these are empty lacunae seen here, whereas in blue, these are the osteocyte cells that sit within the lacuna. The lamellae, these are now each of the circular rings, and this model makes it clear. It highlights from yellow to white to make it really obvious. So each of these layers here specifically would be concentric lamellae, or just simply lamellae labeled here. And then taking a closer look at a single lacuna, you can see the canals that branch off of it. And this has a better perspective. This is the appearance of the canaliculi. And it really means tiny canals that branch off of a single lacuna. So here's a really good example, seeing multiple canaliculi and the lacuna they connect to. And to finish this lab 6, it goes into the histology. So starting really similar to that model, because that's exactly what it was here, we have now the compact bone histology, and we're going to see those similar terms. So the compact bone sample, and you can already make out now the circular cross-section appearance here of a single osteon. And in the center of the osteon, there's a central canal. Surrounding the central canal here is all these multiple dark spaces. These are all the multiple lacunae. We're not able to see osteocyte cells on this preparation, but we can see the spaces that are filled in with black ash, and these are the spaces of the lacunae. And what appears is, of course, the black lines that branch off of them. These thin black lines, these are the multiple canaliculi. So a view of the compact bone histology. Next, there's a spongy bone sample made of the trabeculae. And on this sample, we can see various bone cells. So we'll go here. So to start out with the trabeculae, that's what we see stained in pink, and we'll highlight that. So this entire fragment right here, and anything that has that similar appearance, you can see up here another trabecula. So this entire pink section here, that's the bone of the trabecula. And now embedded within the trabecula, seen here, these purple dots embedded inside, these are osteocytes, the mature cells. So the osteocytes, whereas the purple cells bordering right on the edge of the trabecula, so we got to look at location now. The purple dots represented here are truly just the nuclei of the osteoblasts. So the young, more active cells. Osteocytes embedded deep inside the trabecula. Osteoblasts on the border of the trabecula. And now the red bone marrow, we're looking in the area between trabeculae. You can recognize the appearance of adipose tissue, means we're looking at yellow bone marrow, but specifically the pink clusters, that's where we're seeing the red bone marrow. And you can see various clusters of red bone marrow found between the adipose tissue. And a closer look at this sample, so we're zoomed in. Again, you're seeing the red bone marrow. For reference, I just want to point out all the adipose tissue you see on this spongy bone sample. We have this pink fragment here of the trabecula and osteocytes embedded inside, and osteoblast cells right along the border of the trabecula. The next sample we're going to now is dense regular connective tissue, and this is coming up in the skeleton due to the fact that tendons, ligaments are composed of dense regular connective tissue, a dense parallel arrangement of collagen fibers, and the fibroblast cells that produce them. So that brings us here to this sample. So all of that strength of collagen, our tendons, our muscles connect on via tendons onto the skeleton, or ligaments bone to bone. But again, all of the pink material see here is a dense bundling of collagen fibers. And you can see between the collagen bundles, you can see the location of fibroblast nuclei. And if you were to label it fibroblast or fibrocyte, that would be correct either way. That'd be acceptable. So now we're going to move into another type of cartilage, fibrocartilage. And the reason being now, fibrocartilage is found in the skeleton, the pubic symphysis, the cartilage connecting the pelvis structure together, and the intervertebral discs between our vertebrae. Those are some examples of fibrocartilage that's in the skeleton. Also, certain joints contain pads of fibrocartilage inside of the joint. So this time we're going to see collagen fibers. However, they're now going to be stained in blue. And we're going to see cartilage producing cells, the mature chondrocytes in the chamber of a lacuna. So here we are with the sample of fibrocartilage. And again, samples have been stained so that we can highlight certain features. And it turns out in this case, collagen has been stained in a blue color. So we can recognize this one here. Just a large amount of blue, kind of looks like that wavy appearance. That's the appearance of our fibrocartilage sample. And we can see now the bubble, that's the space of the lacuna. And the dark spot inside would be the nucleus of the cartilage producing cell, the chondrocyte. We just have this sample here for our fibrocartilage. So a stronger form of collagen, a stronger form of cartilage with the abundance of the strong collagen fibers. These last two samples now, we're looking at ossification. So it's showing the process of bone development. So these samples were taken from a specimen that was still in the developmental stage, building bone structures. And we're going to notice here, these samples have trabeculae, osteoblasts, osteocytes on each of these. And it's that same terminology that we found on spongy bone. And it really makes sense because these two samples are still in the process of forming mature spongy bone. So we see some similarities. So of course, we're going to point out what's different. How could we recognize each of these? So intramembranous ossification, we'll bring that up right here. So once again in pink, this entire pink fragment, or you see a larger one here, this is a trabecula. It's just a very small developing fragment of a trabecula. And again, anywhere you see trabeculae, the osteocytes are deep in the center. The osteocytes are the mature cells that have already surrounded themselves in matrix, so they're embedded in trabecula. And the cells right along the border, once again, osteoblasts. If this was still a living sample, osteoblasts would produce more bone, and that would extend the size of the trabecula. And once that happens, these osteoblasts, surrounded in matrix, would mature to become themselves osteocytes. So now the big difference is now looking completely outside of the trabecula. So say in this area, you notice we don't see the red bone marrow. We do not see the adipose. Instead, we have just the undeveloped connective tissue stem cell. We just see a big cluster of mesenchyme. And that's the big difference. When you see a spicule just surrounded by mesenchyme, this must be intramembranous. And if we just go back, when you see a spicule surrounded by adipose, this must be spongy bone. So now our last sample, intramembranous ossification we just viewed, and now going into endochondral ossification, the cartilage type of development. You notice on this sample, we have kind of a wider zoomed out view. And on one side of the view, we're seeing cartilage. That's this view here. This is the hyaline cartilage that will be ossified. And within the cartilage, you notice as we move closer and closer to the developing bone, and we'll just label that. This side is the developing bone with these developing trabeculae, and this is the cartilage side. As we get closer and closer to the newly created bone, this is where we have the hypertrophied chondrocytes. So I'll just put HC. And hypertrophy is just a word for swollen, enlarged. So as the bone's developing and ossifying and calcifying the area of the cartilage cells swell up in that area and then, of course, die off. Whereas in contrast, further away from the ossification region, you just see the normal-sized, healthy chondrocytes. But we want to view the enlarged chondrocytes. So again, it's in this area is where we see just the newly developed trabeculae of spongy bone with all of the familiar components, osteoblasts and osteocytes. So we're going to zoom in on the bone portion specifically. But to do that, imagine we were just going to scroll through the slide over in this direction. So further away from the cartilage here, we see just the larger, more developed trabeculae. And again, as usual, osteocytes in the center and osteoblast cells right along the border. Here's a sample of compact bone using this virtual microscope. And you have the link for this website. So starting out, we can see multiple osteons. You just got to look for that cross section of the cylinder. And then we'll now just zoom in. And that's what we're able to see here, this area of osteon with its central canal spanning around the multiple lacunae. And we'll zoom it in a little closer. Here's a very nice view of now the canaliculus, plural canaliculi, multiple canaliculi that connect to that lacuna. Here's an example of the intramembranous ossification sample. And that's what we can see here. Zoomed out, you point to the pink area of a trabecula. So, of course, the cells embedded deep within the trabecula. These are the osteocytes with the osteoblasts right along the border of the trabecula. And if we zoom out, we can see many examples of those. And especially when we zoom out, we see that lack of adipose. It's another indication that we're seeing the endochondral, the intramembranous ossification. And instead of adipose, we're seeing just a collection of the mesenchyme cells. Here we have a sample of endochondral ossification. And we can see down here at the bottom of this sample is where we see the cartilage portion. In fact, looking at the cartilage, we can see here the area of the hypertrophied, the enlarged chondrocytes, the really big ones appear like stacked up in their appearance. And then we get further away again, we have the normal size cartilage cells. And then moving to this area, we see kind of this darker, kind of reddish color, the staining for these trabeculae. And as usual, zooming in closer, we have the cells bordering the trabeculae. osteoblasts, and the ones embedded deeper inside of the trabeculae, the mature osteocytes. But again, for this sample to recognize it, we want to zoom out so that we see partial developing bone, spongy bone here, and then partial dying hypertrophied cartilage. So now we're going to move into the Lab 7, relatively short section, just looking at the types of joints. So we want to start right here. There's a category of fibrous joints. There's 3 specific examples, and we're just pointing to locations where we find these. And we'll use our poster for this. So the suture is the name for the joints between our skull bones. So we could see that here, just these major joints right along the skull. So where the frontal and parietal bones meet, between frontal and temporal. Those are just some examples of the major sutures along the skull. And we'll continue on. A syndesmosis joint, just as it says here, just a typical ligament. Whereas gomphosis, these are the unique ligaments that connect our teeth into their sockets. So once again, we'll just go back to the poster. And we can see right here at the shoulder, for example, an example of those typical ligaments. Got syndesmosis, like here at the shoulder. Whereas here, just pointing to the teeth right there at the root of the tooth, that would be the location, the only location for a gomphosis joint. We don't actually see the ligaments from this view. Cartilaginous types of joints, and the 2 specific examples, synchondrosis or symphysis. And we'll once again point out their locations. A synchondrosis, meaning it's composed of hyaline cartilage. The costal cartilage between the ribs and the sternum are a good example of a synchondrosis joint. Whereas a symphysis joint, here we are viewing the intervertebral discs. These pads made of fibrocartilage, as well as here the pubic symphysis, connecting the 2 sections of the coxal bones together. That pubic symphysis, also made of fibrocartilage, an example of a symphysis joint. And then lastly, a synovial joint, the most common based on these categories here. Synovial joints, and we just point to the elbow joint, the shoulder joint, the hip joint, an extremely common type of joint. And in fact, the further details of synovial joint are seen on this figure, 8.1, and we'll go through the detailed features. And we'll just go ahead and bring that up right here. So on this figure, we're looking at a synovial joint. The synovial joint itself is held together by this articular capsule, and it has its 2 subcomponents there. The superficial fibrous joint capsule, that's seen here in tan, and the deeper synovial membrane, the source of our joint fluid, our synovial fluid. So the capsule is what holds the bones together at the articulation. And another key trait of synovial joints, it contains this liquid-filled joint cavity. The synovial membrane is the source of the synovial fluid that fills this space. And a remnant of our endochondral ossification. Here's that protective hyaline cartilage left at the ends of the bones, and it's known as articular cartilage. So that protective pad right there at the ends, and we see that facing into the joint cavity. So any of that detail seen right here on